Today we're going to look at the second part of chapter 6. We're going to look at intramembranous ossification, and also we will be exploring endochondral ossification. So the first one we're going to look at is going to be the intramembranous ossification. This is also known as dermal ossification. We always see dermal ossification occurring in the flat bones of the skull. Flat bones are also located in the mandible and also the clavicle. So the flat bones are going to have three distinct layers. The outer layer is going to be compact bone, and then an inner layer of spongy bone, and then another layer of compact bone. So the first step in intramembranous ossification is usually occurring about the 8th week of development. We're going to particularly pay attention to the mesenchymal cells. These are going to begin division, and remembering that one of these mesenchymal cells will become another mesenchymal cell, and the other becomes a committed cell, which will then eventually become an osteoprogenitor cells, and some of the cells are going to then become osteoblasts. This collection of osteoblasts is called an ossification center. In step 2, we see the main production of osteoid. So osteoid is produced by the osteoblast, and it's a matrix which contains nutrients for the osteoblast. And this osteoid formation will be then followed by calcification, and it's showing it here. So this calcification is usually first in the form of calcium salts, which are deposited inside of the ossification center. We will also begin to see the osteocytes, which osteocytes are mature bone cells, which are going to then receive all of its nutrients from blood vessels. In step 3, some particular areas that we're going to look at first is going to be the mesenchyme. So the mesenchymal cells are going to aggregate or are condensed to form the outer layer, which is known as the periosteum. So we're going to see a series of mesenchymal cells aggregating towards the outside. Again, we will see the division, and the division of these are going to form an osteoprogenitor cell or an osteoblast. What we also see is the beginning of blood vessel integration. So the blood vessels are going to start weaving into an unmature bone called a trabecula or trabecula. We also see osteocytes, osteoblasts aggregating or collecting along the trabecula. And we also see again the mature osteocytes, which are then going to receive all of its nutrients eventually from blood vessels. And finally, in step 4, we see two different components. The first one is going to be the periosteum. So we can see two things that are happening. We see osteoprogenitor cells, which then can divide into an osteoblast, or in some cases we can also see it divide into an osteoclast. And we also see the mesenchymal cells, which are still lining up along the periosteum. These osteoblasts are important. They're laying down a little bit of the bone matrix on the outside. The second stage, what we see is the formation of a lamellar bone. And the lamellar bone is going to be two components. So we're going to see the division or the compact bone, which is formed on the outside. Then we're going to see a layer of spongy bone, which we can see now the trabeculae, they're shadowed in brown here. But it's a lattice framework where all the osteocytes are going to lie, receiving all their nutrients from the blood vessels. We also see the osteoblasts, which are important. These are eventually going to transition to the osteocytes. And then we see another layer here of compact bone, and then finally we see the periosteum, which has the osteoprogenitor cells and the osteoblast located on the outside. When we're looking at endochondral ossification in particular, we're really looking at changing fetal hyaline cartilage into eventually bone. So in step 1, we usually see this occurring anywhere between about the 8th to the 12th week. So the first thing that we see is the fetal hyaline cartilage model starts to develop. Inside of the fetal hyaline cartilage, we have chondroblasts, which are secreting cartilage matrix, which helps form the hyaline cartilage model. We also see chondrocytes that are going to be trapped in their own individual lacunae. Here's a picture of one right here. So each one of these are still located in step number 1 inside a lacunae. And we do see a very thin perichondrial layer that gives the fetal hyaline cartilage some resemblance of structure. In step 2, we begin to see the chondrocytes start to enlarge and hypertrophy. So that's represented with these white lacunae with the chondrocytes located inside. So they start to hypertrophy and reabsorb the cartilage matrix. This will cause the cartilage matrix to calcify eventually. These chondrocytes are then going to begin to die because they were receiving nutrients through the matrix and now are unable to diffuse into the cell. We also see the blood vessel. So that's what this is on the left side of this picture. So we have blood entering through the perichondrium into the periosteal bone collar. Once the blood vessels begin to penetrate the perichondrium, we'll see stem cells within the perichondrium dividing to form osteoblasts. These osteoblasts then will secrete osteoid, and eventually the osteoid will harden to form the periosteal bone collar. In step number 3, we classify this as the primary ossification center. And within this primary ossification center, we're going to see a couple of increases or new develops occurring in the diaphysis. So the first thing that we note is we're going to start seeing an increase of blood flow. An increase in blood flow means that we're also increasing osteoblast activities, extending from the periosteum into the diaphysis. So the more blood flow we have, the more osteoblasts that are going to migrate into the diaphysis. These osteoblasts will secrete secretions of osteoid inside, and eventually the osteoid will harden to become bone, which gives us the name the primary bone ossification. This is the first area of bone formation in endochondral ossification, and bone formation is going to not only extend upwards but also downwards towards the epiphysis in step number 3. So we're starting to begin to see the bone growing in length. In step number 4, very similar to step number 3, we call this the epiphyseal or the secondary ossification centers, which are occurring in the epiphysis. So just like in step number 3, we see an increase of blood flow, and these increase in blood flow will bring the osteoprogenitor cells, which will eventually change into the osteoblast. And then we'll see those osteoblasts laying down osteoid, which will then become bone. This secondary ossification center is basically replacing cartilage with bone. In the diaphysis, what we're going to see is the formation of the medullary cavity. This formation of the medullary cavity is due to the increase of osteoclast activity. Osteoclast, as you remember, increases the reabsorption of the bone matrix, and that helps form the medullary cavity. In step number 5, we see that bone has replaced the majority of the cartilage. So we can see in the diaphysis, we have the bone with the medullary cavity, and in the epiphysis, we have the trabecula or the spicules of spongy surrounded by compact bone. There are 2 areas that we see an exception. First is the articular cartilage, or otherwise known as hyaline cartilage, which is located on the proximal and the distal articulating surfaces of this long bone. And also we see this region right here between the epiphysis and the diaphysis, this metaphysis area, a thing called an epiphyseal plate. And we can still see the individual chondrocytes located in their lacuna. Eventually what we're going to see is this epiphyseal plate. This individual is continuing to grow in length. And we'll see when these epiphyseal plates are closed, it means that these cartilage cells have been replaced by bone cells. So in step number 6, we see the first part is going to be this epiphyseal line. And this epiphyseal line, I'll draw it down here. This means that we are now finished with lengthwise growth. And we also see an epiphyseal line located in the proximal surface. So at this point, we know that this individual will no longer grow in height. And after this stage, what we'll see is bone development occurring with bone remodeling, which means that we're going to grow in overall diameter. So once these epiphyseal plates are closed, we consider step number 6 to be adulthood. And that concludes the lecture on ossification processes in flat bones and long bones.